we finished talking about the dihybrid cross. And before we move on to the dihybrid cross, it's important to understand how groundbreaking the monohybrid cross was for Mendel. Mendel was able to show that these tall plants had characteristics that were inherent to these tall plants, and that these short plants had characteristics that were inherent to the short plants. He cross-breeded them. I mean, he took the pollen of one and mixed them with the ovary stamen of another, or the pistil of another. And he ended up with offspring that were all tall. Now, this was, interesting to him. Imagine how, okay, great, we now have tall plants. And then he bred these tall plants with each other, and he found out that some of them were short. Now, something that nobody had, people were doing this on their own. Like, if you wanted the best apple tree in the world, you would take the characteristics of the apple you wanted, and you'd grow those seeds, right? But that's good in what he was doing, but he was looking at it from a kind of a scientist, geneticist, hereditary perspective, and he found that there were characters in this plant. There was an allele in this plant that of shortness that was being masked somehow, and that came back in the second generation. So something was here that wasn't showing itself, but it was there, and it was being passed on to the next generation. Now these he called them invisible units. That the short thing was invisible in a unit in here, but it was still crossed. It was still passed on to the next generation. That is groundbreaking. Nobody really knew what was going on with heredity. Now, Charles Darwin was saying evolution is this and evolution is that, and people were trying to find different mechanisms for evolution, but nobody knew what Mendel knew. Nobody. And it wasn't until about 50, 60 years later that people found Mendel's work. And Darwin, if Darwin had read Mendel's work, Darwin would have known the mechanism by which evolution happens. Darwin knew that evolution occurred, and he had tons of evidence for it. But he had no idea how things were passed on from one organism to another. And this is what Mendel had. This is what Mendel knew. All right. So the monohybrid cross is super important. Now the dihybrid cross isn't really that much different. He's just using two characters now instead of one. All right. So instead of using just tall and short plants, he's using tall plants with purple flowers versus short plants with white flowers. All right. And he observed their growth. Now, he was trying to find a few things. Now, he wanted to know if these characters assorted dependent of another or independently of one another. Meaning, do the Y's and the R's, are they being passed on from one generation to another with one another? Does the R's and the Y's, are they being passed on independent of one another or dependent of one another? All right. So if one was dependent on the other, then the big R's and the big Y's would tend to stay together, and the little R's and the little Y's would tend to stay together. All right. So that was one hypothesis. The other hypothesis is that they independent assortedly, which is actually what his results ended up showing. So I hope you can see this here. I don't know if it's clear or not, but one organism in the dihybrid cross had all the dominant alleles. All right. So remember, the R's are one character and the Y's are another character, and there's two alleles of each character. So what we have here is a true breeding round and yellow seed plant with a true breeding wrinkled and green seeded plant holding the recessive phenotype. Okay. Now he crossed them and he wanted to see what would happen. All right. Remember, he also wanted to know if we know that these characters, are going to be sorted dependently on one another because they're in the same place in a homologous chromosome. He wanted to know what is the interaction between the two. Okay, so here's what he did. he ends up with a F1 generation of all round and all yellow plants. He ended up with plants that produced seeds that were round and yellow. Okay? And this is not really different from the monohybrid cross, all right, in that all of the F1 generation were heterozygotes displaying the dominant phenotype. Again, we have heterozygotes displaying the dominant phenotype. The crazy thing happened when he took one of these, a plant with one of these, and a plant with two plants with this same phenotype and genotype mixture. And remember, he didn't know the genotype. He's just looking at phenotypes here. All right. He cross fertilizes these two plants, and he ended up finding something called a 9:3 to 3:1 phenotype ratio. All right. Remember, it's important for you to know we only do ratios for phenotypes. We don't do ratios for genotypes. We don't count up genotypes. We count up phenotypes. So this phenotype is 1, 2, 3, 4, 5, 6, 7, 8, 9. Had 9 possibles out of 16 had a double dominant phenotype, dominant round, dominant yellow. 3 of them had a dominant phenotype of being yellow, but a recessive phenotype in being wrinkled. And 3 of them had a dominant phenotype of being round, but a recessive phenotype of being green. All right. And 1 of them had both recessive phenotypes. And he found something like, 950 to 322 to 292 to 114. He didn't find exactly 9:3 to 1, but he found a general 9:3 to 1 ratio. Every time he did this F1 cross, he found a 9:3 to 1 ratio with his offspring. Okay, so this was the dihybrid cross. What this showed actually was that the Ys here did not assort during meiosis. They didn't separate with the big Rs. They separated completely independently of one another, which means it was just as common to get a big Y, little y with 2 big Rs as it was to get a big Y with little y with big R, little r, as it was to get a big Y, little y with 2 little r's. So what this showed was that during the process of meiosis, this character, color, was not assorting with another character, shape. Okay? Not happening. Okay, so 2 things. Segregation and independent assortment are the 2 major laws, or 2 major things that Mendel proved as being existent within the inheritance of characters and traits. All right. One of them, the law of segregation. All right. Alleles separate during processes of gamete formation. Now that makes sense. Okay. So here, law of segregation. Imagine that we have a chromosome, all right, that duplicates during the S phase. Leaving us with a tetrad. Now, during the process of meiosis, we have 1 division. All right, and when crossing over occurs, we're not going to talk about that for now. What's going to lead to one group being small a, and the other group being, big A, big A. Now, what do we know about meiosis? What do we know about meiosis? What's going to happen next? Remember, we have another division to go through, right? We have another division. So during our next division, one of them, big A, another one, cell will get big A. For these 2 will separate. One cell will get little a, and another cell will get little a. So we're going to end up with 4 new cells, 1, 2, 3, 4, all having one of these alleles. Only one of the sperm, or one of the eggs, will get one of these alleles. All right? So this means that during the formation of gametes, all right, the alleles will separate throughout the gametes, all right, and even though we started out with a cell that had a big A allele and a little a allele, they are going to separate during gamete formation. You will never inherit that from a parent. You can only inherit one or the other. Now we move on to independent assortment. Now, independent assortment says is that different characters or different alleles of different characters are actually being divvied up throughout the gametes separately of one another. For example, let's say that big A, small a has to do with eye color, and that big T, small t has to do with height. So now we have two characters, all right? Now, these characters, you have 23 chromosomes, we have 23 pairs of chromosomes with 46 chromosomes. It's likely that these two characters are not on the same chromosome. We're going to talk about what would happen if they are in the same chromosome. They might not sort independently of one another. But let's focus now on the fact that these two characters are on separate chromosomes. And so what's happening is when we're producing gametes, these traits are sorting independently. Now, here's the crazy thing. The possibility of this A going this way, all right, during cell division, you know, and this one going this way, all right, is not related to the possibility of this T going this way and this T going that way. Now, what's dependent is these. This is dependent. If this A goes this way, this A is going to have to get pulled the other way because they separate, right, after metaphase. But the possibility of this A going this way and this A going this way has nothing to do with the chance that this tetrad, or this pair here, is oriented just a little bit differently at the metaphase plate. And so this T might get pulled this way, and this T might get pulled in that direction. So what this is saying is that the separation of these alleles during gamete formation is not dependent upon the separation of those. And the separation of these, whichever way they separate, has nothing to do with the way that these could possibly separate. And that is why there's so much genetic variation. Because this is happening with 23 pairs of chromosomes that have all duplicated into tetrads and crossed over and are now separating throughout 4 gametes from one cell. It's crazy, I know. So we talked about law of segregation and law of independent assortment. And that is basically the law of independent assortment focuses on the idea that one character on one chromosome separates independently of another chromosome. And luckily for Mendel, with his pea plant experiments, all right, these were actually on separate chromosomes. All right, but what if, and we have a chromosome here, all right, and this is the centromere region, all right, what if we had 3 different characters on one chromosome? How do you think they might be inherited together or separately? Like, if A, B, and T are all characters or all alleles of characters that are on the same chromosome, you know, this whole thing is a sister chromatid. They're on the same chromosome. What are the chances that A and B are inherited together? Versus what are the chances that A and T are inherited together? And what are the chances that B and T are inherited together? All right. So one of the questions for this slide, and it's the last question that you have for this video, is I would like you to try to answer right now in your head, are the chances of A and B being inherited together greater or less than the chances of A and T being inherited together? Are the chances of B and T being inherited together greater or less than the chances of T and A being inherited together?